Hello my dear children, Namaste and welcome to yet another session of our newly launched ICSE Class 10 Biology series. I am Ambika, Master Teacher of Biology right here at Vedantu. Now you must be wondering why I keep saying ICSE, right? It's only because our preference right now is to do a little bit of extra justice to the ICSE children, because all these days we've done a lot for CBSE and we are still continuing to do a lot of wonderful things for the CBSE children. But even if right now our syllabus is focused on ICSE, I know that a lot of you are from CBSE and state boards, so just keep an eye on the topic that we are discussing in the sessions, just in case they are one of the topics that you also have, like overlapping topics between CBSE, state boards or ICSE, whatever syllabus it is. If it is a topic that you have to learn as well, please do make it a point to attend the session, right? I'm sure it will help you because the more you hear about a topic, the easier it becomes to retain in your memory, right? Even if it is for lower grades, because some children have been asking me, ma'am, can you do sessions for 9th grade? Right, so there are some 9th grade syllabi in the country, like I think some CBSE schools and some state boards have topics like mitosis, meiosis and all of that in 9th grade. So even if you are from a lower grade, if at all you find this topic relevant to you, please definitely check the schedule for the day and make it a point to attend the session. Right? So let's get started. As you know, we have already done the chapter cell cycle and cell division, the major topics from that, which were the cell cycle in our first session. Our second session was about mitosis, which all of you really wanted to have a session on, and our third session was on meiosis, which is a little less focused on the ICSE syllabus, but it is still part of your syllabus, right? So I did make it a point to elaborate a little on the process, just so that it becomes easier for you to understand genetics and human reproduction, which will be chapters that we take up as we go along in the weeks to come. Okay, so today, as you can see, the topic that we will be discussing is structure of chromosomes. So this, in some of your textbooks, it is part of the same chapter, cell cycle and cell division. In some textbooks, it's discussed as a separate chapter, but doesn't matter as long as this topic is part of the syllabus. We need to understand all that is in it, and that's all that matters, right? So let's get started. But before that, we have a task, right? The homework question that I had given in the previous session: what are the major differences between mitosis and meiosis? I was glad to see that I got some very quick answers. I have put in 2 answers that came first. One was from Shashi, who says, mitosis gives 2 nuclei and hence 2 cells, while meiosis gives 4. Mitosis gives identical cells to each other and to the mother cell, while meiosis leads to genetic variation due to crossing over and independent assortment. Mitosis gives nuclei with the same amount of chromosomes as the mother cell, while meiosis gives cells with half the number. Mitosis includes 1 division, while meiosis includes 2. I think Shashi has done a wonderful job of explaining. It in fact shows the sincerity in you, that you have understood the concept, you have taken pains to understand both the concepts, and you have in fact compiled this answer. Thank you very much, Shashi, and do keep it up. And we have one more answer from the ID says Vikas, but the name here at the bottom says Kanishk. Thank you very much, Kanishk. Kanishk says in mitosis, the number of chromosomes remain the same. It occurs almost in our entire body. It leads to 2 daughter cells, which is very correct. And meiosis, the number of chromosomes is half of the actual one. It occurs in our gametes and it leads to 4 daughter cells. And thank you very much, Kanishk, for loving the session. Just like Kanishk, in case you have also enjoyed and benefited from the sessions that I have taken so far, do not forget to hit the like buttons right now and do share it with your near and dear ones, your juniors if you think it's relevant to them, your friends of other schools, other boards if you think it will be useful to them, do share it with them, and do not forget to subscribe to our channel Vedantu 9th and 10th, because as I keep saying, we have been and we are still continue to work very hard to give you a lot of amazing content just so that your board exams and your final exams can be much easier for you to write and come out with flying colors, right? So stay tuned and let's get started with today's session. Now, this is an interesting fact that I found. It's a very simple thing, but most of us are not aware of this. The human body, in fact, is made up of approximately 40 to 50 trillion cells. Now, how much is a trillion? Can anybody tell me? Put it in the chat box there. I will be watching your comments as the session is being premiered. Put it in the comment section. Otherwise, if you're watching it later, okay? How much is 1 trillion? And the human body is supposed to be made up of around 40 to 50 trillion cells. Wow, that is so much. And to imagine that we came to that from one single zygotic cell is just amazing. Imagine the extent of cell division that our body would have gone through, right? So many rounds of cell division, so many mitotic divisions would have led to what we are today, right? Now, one more thing, which is a good to know fact rather than fun, honestly speaking, I would call it a good to know fact. This is something that you know already. The genetic material in the nucleus of each cell is seen as thread-like structures called chromatin. So the picture that I have here is the nucleus of an individual cell. So I want you to visualize it like this. The human body, the first image that we saw, the human body made up of trillions of cells. Just close your eyes and visualize that you are zooming into that, and you are zooming into one individual cell of the human body. Inside the cell, you zoom into the nucleus, which is the most important part of any typical cell, correct? That is what I have on screen here. This is roughly what you would see inside the nucleus. You have the nucleolus, you have the endoplasmic reticulum, which is right outside the nucleus, and the nucleoplasm lying all around inside the nucleus, the fluidy part of the nucleus, the nuclear pores. Here, the openings, the tiny pores or the openings that you see, and the nuclear envelope, which is the membrane that surrounds the nucleus, the double-layered membrane that surrounds the nucleus. The most important part in the nucleus is definitely the genetic material, which is seen as thread-like structures called chromatin. Right? This is what chromatin is. The thread-like structures that you see here scattered all over. Chromatin. Children, keep in mind these terms. They are very important because we have been discussing them, and today I thought it's useful to have one individual session in this chapter regarding these terms, because only if these terms are clear in your mind, we will be able to progress smoothly with chapters like human reproduction and genetics, which will be taken up in the weeks to come, right? So chromatin is this form of scattered thread-like structures that you would see in the nucleus of a cell, especially during interphase. Interphase is when you would find a cell having chromatin lying around in its nucleus in this form, right? So again, just to emphasize on that and to bring back what we have done in the previous sessions, I have put these two images. This is a picture of a cell in interphase. Like I said, you can see the chromatin here. By the time a cell completes interphase, remember G1 is G2, right? And by the time a cell completes G2, it is ready to enter prophase of mitosis, right? And when it enters prophase, during prophase, what happens is these thread-like chromatin or thread-like structures condense into the X-shaped structures that you see here. Well through prophase, or at least towards the end of prophase, this is how a cell would look like. And at this point of time, you wouldn't call this chromatin anymore. You would in fact give it a different name, which is chromosome. Okay, because they are arranged as more systematically seen thickened condensed bodies rather than a random thread mass. Okay, so chromosome and chromatin. I hope you have got the difference between the two. Let's now go ahead. Now, talking about the discovery of chromosomes, it was this scientist, Walter Fleming, who in 1882 discovered chromosomes in the rapidly dividing cells of the larvae of salamander. Now, salamander is an amphibian, and he was studying the cells of those rapidly dividing cells of the larvae of salamander, and happened to observe certain thread-like structures in the cells, which he identified as chromosomes. And that was when chromosomes were first discovered. But then Fleming did not have too much of an idea about the organized structure or the random thread-like mass or anything like that. To him, they just appeared as a thread, and the first time he named them, he calls them chromosomes. And why did he name them chromosomes? There is a reason to that as well, because these structures, they take up certain stains or dyes very easily when they are stained. Okay, for the purpose of experiments in a biology lab, different kinds of stains are used, right? You must have learned even in your practical classes, right? So since these structures take up some of the stains very easily, they appear as colored structures. And that is how the name chromosome came into existence. Chromosome, because chroma means color and soma is body. Colored bodies, right? Which is why he calls them chromosomes, right? Where have you heard the suffix soma before? Can you recall? Well, we discussed this in yesterday's session. We talked about gametic cells and somatic cells. Somatic cells are casually called body cells, and we refer to them as any random cell of your body apart from your gametes, right? Somatic or body cells. The same thing here. Soma is body and chroma is color. So colored body, chromosome is the name that we have been commonly using even today, right? But the only thing was that because of the lack of availability of high resolution microscopes around his time, Fleming was able to observe only the random chromatin structure. It wasn't exactly more of the detailed structure that we know about today. All those discoveries came up much later. Okay, so this is briefly about the history of the discovery of the chromosome. Now, coming to the structures that we know, I'm sure you know this, but let's just quickly recap that once again. Now, this structure, the first one that you see here, is what a typical chromosome would look like. What is this structure called? Would you happen to remember? It is called the centromere. Okay, it's called the centromere. And what are these? They are nothing but the arms of a chromosome. They are just called arms. Okay, these two. The two parts that you see on either side of the centrosome. Now, what is the difference between this and this structure? This is also a chromosome. Some of us even well relate to the word chromosome when we look at this structure rather than to this structure, right? That is because at the time of cell division, once the DNA has duplicated, the chromosomes end up taking up this form, wherein the duplicated DNA gives it this, and we call them sister chromatids. Okay, these two are sister chromatids held together by the centromere again. Okay, so what I'm trying to say is that both of these are chromosomes, the only difference being this is seen only at the time of cell division after the DNA has duplicated, and this is how the typical chromosome structure is supposed to be. Okay, so do not get confused. In this structure you have two sister chromatids. Here we don't differentiate it as sister chromatids, it's just two arms, right? And here the sister chromatids are held together by the centromere. What other function does the centromere have? If you have seen our session on mitosis and meiosis, of course, you would recall that the spindle fibers, right, which the centrioles give out at the time of cell division, attach themselves to the centromere. And it is after attachment to this part that the sister chromatids are pulled away from each other. So centromere is a very, very important part of the chromosome. So chromosome, chromatid or sister chromatids, right? So chromatin, chromosome, chromatid. I hope you have these three terms clear in your head. Now let us look at this. Now this is a very, very interesting fact. Okay, now what do you think I have here on this side of mine? No, it's not a bowl of noodles, even if it looks like that. This is how chromatin looks. This is what chromatin looks like inside. If you look at a 3D model of the nucleus, okay, these are the nuclear pores, and the yellow mass that you see here, which looks like noodles, is chromatin, the network chromatin. And just read the facts that I've written there. If you unwound all the DNA in a single cell, it would form a string that's around 2.2 meters long. Wow, I don't know, it's so hard to imagine, because just think about how long is 2.2 meters. That means it's 220 centimeters. Well, I think my height is approximately 1.68 meters. So one single cell's DNA, imagine one cell, you can't even see the cell with your eye, keep that in mind, with your naked eye. Imagine inside the cell you have so much of DNA, so long that if you just pulled out this thread of noodles and made it stand next to me or hung it next to me, it would be much taller than me, around 220 centimeters. Wow, that is a lot. So another fun fact that I have is all the DNA strands from one single person, if unraveled, would form a string that's equal to 300 times the distance between the sun and the earth. What do I mean by that? If you, I said that the human body is made of around 40 to 50 trillion cells, right? If you were to take out the DNA of each of those 50 trillion cells, laid them end to end like a long railway track, it would be very, very, very long. It would be so long that it would be 300 times the distance between the earth and the sun. Imagine the magic of genetics. Imagine the magic of biology. How can that much be packaged into one single body or one individual cell of a body, which is such a tiny microscopic structure, right? So to understand that, we will have to see how this is done. How does the cell manage to do all of this, right? How does the tiny cell manage to do so much? We really need to know how it manages, because I know that, and I have seen people also, when you have a trip, that too when you have to stay, take a week long trip, and you have to pack your bags, we find it so hard to travel light, right? Most of the time I end up carrying like 2-3 bags minimum, probably because I am not a packaging expert as good as our cell is. Perhaps we have to learn a lot of packaging techniques from our cell. Let's see how the cell manages to do it. Now, this is how it is. It may look like a weird structure that you may not have seen in biology before, but it's a very interesting thing because of how organized it works. Okay, so what is chromatin made of? It's important to understand that. We know it's a thread, we know it is the genetic material, but basically what is it made of? The chromatin is made up of DNA and histone proteins. What is DNA? DNA is deoxyribonucleic acid. Biochemically, that's what the genetic material is in our body. Histone is a type of protein that's found in our body. DNA and histones are together formed into different clusters and arranged into the thread that we know as chromatin. Now, here I've just put this is not part of the cell or anything like that, but you must have seen packaging rolls like these, right? Like those packers and movers, when they come, if you have ever shifted, you would certainly have seen a lot of rolls like these. That is what packaging experts use. So who are our packaging experts? Our cells packaging experts are in fact the histone proteins. Now you can see this is the chromatin that I pull out of the nuclear pore of the cell and zoom into it. This is the DNA. This is basically the DNA. It's made up of DNA. That's what I'm saying. These are histone proteins. The bead-like structures that you see are the histone proteins. You see how coiled the DNA is around those proteins, right? Here it's much more clearly seen, right? Because of this coiling and organizing, it is able to pack in meters and meters of genetic material into one single cell. Now let's look at another picture with a few more labels. We have the chromatin inside the nucleus. We have the nuclear pore. I pull out a chromatin fiber through the nuclear pore, and you can see that I'm winding it around the histone proteins. This is the DNA helix. If I zoom into it, basically it's made up of DNA, which is what is coiled around the histone proteins. And ultimately, this is what the condensed form of the chromosome looks. Okay, chromatin is this. After condensation, the X-shaped structure with sister chromatids looks like this. So what you can see here is that it's basically the same noodles thing that makes this also. The only difference is that it's organized into systematic shapes like these during cell division. Okay, now coming to a little more details on those, because I know it looks like a messy mass, all of that packaging there. Now histones, I've put this here for your information. Histones are organized to form a unit of 8 protein subunits to form a histone octamer. Now what do I mean by that? An octamer, octa means 8, octagon is a shape which has 8 sides, right? So octamer has to give you the clue that it's made up of 8 subunits. So just keep this in mind. If you're asked what are histones, histones are basically proteins in the body which are organized to form, organized in the form of 8 protein subunits in order to form an octamer. Even if you don't recall the word octamer, that's fine. Just remember that histones, now if you look at it in this particular picture, this is what I am talking about, this structure. You see those individual tiny beads there, right? There are in fact 8 of them, 8 subunits, 8 small beads arranged together to form the histone protein complex, and it is around this complex that the DNA is wound. Okay, so I hope it's very clear. Keep in mind these terms: histones, nucleosome and chromatin, and keep in mind this picture, right? As you can see, it is the long DNA helical structure, as you would call it, the long DNA strand that is wound around. Imagine you have a long thread and you have a round structure, probably this marker that I have here. I'm just winding the thread around this. That is what is happening here. And if I zoom into the structure of this marker, I would see 8 small beads making up one section of the marker, which is what the histone proteins are. And they are packed further to form the chromatin, and finally, of course, at the time of cell division, you see them as chromosomes. Okay? Now, coming to the types of chromosomes, I'm sure you know this already. Chromosomes in general in the human body, especially or in any sexually reproducing organism, are of 2 types, autosomes and sex chromosomes. Now, I'm sure all of you know what these are. Autosomes are the chromosomes in your body that do not determine whether you're a male or a female. Sex chromosomes are those chromosomes in your body or in your cells that determine whether you're a male or a female. So basically, the easier way to understand is knowing what the sex chromosomes are, because a typical human cell has 23 pairs or 46 chromosomes in all, out of which we have 44 autosomes, which we typically represent like this, 44AA, and 2 X chromosomes. Now this is in my case, because for females it will be 2 X chromosomes, and in the case of males it would be 44 autosomes and an X and a Y chromosome in a typical diploid cell. Okay, so sex chromosomes are just the X or the Y chromosomes. So there are just 2 sex chromosomes in a diploid cell of the human body, whereas there are 44 autosomes in the typical diploid cell. Okay, so keep that in mind, what are autosomes and what are sex chromosomes. Again, chromosomes based on centromere. Remember, I told you the centromere is a very, very important part of the chromosome, right? Based on the position of the centromere, chromosomes are further divided into 4 different types. Some of them may not be seen in humans, some of them are seen in humans at different stages. Whatever be it, just understand that based on the centromere's position, chromosomes may either be telocentric, acrocentric, submetacentric, and metacentric. Right, I'm sure we know the meanings of the prefixes meta and telo by now, because we know the stages of mitosis and meiosis. Quickly tell me, what does telo indicate? Quickly tell me, what does telo indicate in telocentric? End, right, or last. So you see that the centromere here in a telocentric chromosome, the centromere is towards the tip of the sister chromatids or the tip of the chromosome, which is why we call it telocentric. The next one is acrocentric, wherein the centromere is slightly away from the tip but still closer to the tip of the chromosome. Okay, slightly away from the extreme tip. Compare these two and you would know the difference. Submetacentric would be the type in which the centromere is slightly away from the middle. Okay, that is what submetacentric is, because meta is middle, which is exactly why in the case of a metacentric chromosome, we say that the centromere is exactly at the middle. So metacentric exactly at the middle, submetacentric slightly away from the middle, telocentric at the tip, and acrocentric slightly away from the tip. Now this is important, children. It could be asked in your exam. Keep in mind the classification or the types of chromosomes based on the position of the centromere. Okay, so I guess we are done with that part. We are done with chromatin. We have understood what chromosomes are. We have understood what sister chromatids are. We have understood how our packaging experts, which are, tell me, the histone proteins, right? How they manage to package the DNA or our genetic material in such an organized and systematic way into a very, very, very organized structure that we see as chromatin. We have understood all of that, right? Now, let us come, So we are also left with one more very small topic, which is the structure of DNA, what is DNA and what are genes, which we will take up in the next class, because as such I didn't want to overload you or overburden you with too many new terms. Perhaps you have done this in your school. Even then, let's take it slowly. I'm sure we have enough time. We will take that up in our next session, okay? DNA and genes. But before signing off for today, let me give you one homework question, which is how many autosomes and sex chromosomes would be found in a human sperm cell? So I'm sure all of you are smart enough to come up with quick answers, but let's see who comes up with the best answer and the quickest answer. And I will be discussing that, of course, in the next class. And of course, do not forget to download Vedantu's learning app and do join our Telegram group so that you always can chat with our expert teachers regarding any of your academic doubts. And we have all of these amazing features out there. And very importantly, children, if you have found this session useful, do not forget to hit the like button. Do share it with your friends. As I told you, your friends of any board, CBSE, state boards, or even 9th grade students, even 11th, 12th, whatever grade you are in, if you think this will be suited and useful for your friends, do hit the share button as well and share it with all your near and dear ones. And if you have not subscribed to our channel yet, please do hit the subscribe button right now before you forget, so that you have access to all our amazing upcoming sessions all the time in the days to come. Because remember, children, we are in this journey together. You and we are always together. Let us aim at doing the best and aiming for the best for your final exams. And in the meantime, if you have any questions, any doubts regarding any of our sessions or anything that I can help you with, do not hesitate to reach out to me at ambika.gopalakrishnan@vedantu.com or put in your specific comments in the comment section below. I'll see you again in another interesting session. Do not miss any of our upcoming sessions. We are coming up with more and more interesting topics in the days to come. So stay tuned. Happy studying and bye-bye.